Liver disease accounts for approximately 2 million deaths per year worldwide, with cirrhosis, viral hepatitis, and malignancy being the most common causes. Consequently, the regenerative capacity of the liver is a topic of extreme interest in the search for curative therapies to end-stage liver disease. Mesenchymal stem cells have emerged as a promising new therapy for hepatic regeneration. MSCs have multiple properties that make them an appropriate treatment option for liver disease. First, MSCs are pluripotent stem cells capable of differentiating into hepatocyte-like cells both in vivo and in vitro. Second, MSCs are readily accessible from multiple potential sources, including adipose tissue, umbilical cord blood, peripheral blood, synovial membranes, muscle, dermis, and liver. Importantly, the harvested MSCs maintain their pluripotent potential, robust proliferative ability, and capacity for ex vivo expansion. Third, MSCs have the ability to migrate to and engraft at the site of injured tissue. Fourth, MSCs have immunosuppressive properties that allow for allogeneic transplantation. The immunosuppressive ability of MSCs also include antifibrotic as well as antioxidant effects, which can protect the liver from fibrosis and oxidative damage. Lastly, MSCs produce extracellular vesicles that contain growth factors and cytokines that promote regeneration of impaired tissue, such as liver parenchyma. In this article, we focus on the potential therapeutic mechanisms of MSCs and future studies that can help develop more effective treatments for end-stage liver disease. A major criterion for effective stem cell therapy is the induction and engraftment of cells into the region of damage. Multiple studies have demonstrated the stromal cell-derived factor 1A, a chemotactic protein of the CXC family produced by bone marrow stromal cells, plays an important role in stem cell homing, chemotaxis, engraftment, proliferation, and survival. The receptor for SDF1A is a chemokine CXC receptor 4, which is found in a variety of cells, including immune cells and hepatocytes. SDF1A is a potent chemoattractant for cells expressing CXCR4 receptors, and studies have demonstrated increased concentrations of SDF1A in the liver following liver injury. MSCs have low native levels of CXCR4. However, genetically modified MSCs with CXCR4 overexpression have showed increased migration and engraftment of MSCs at the liver following acute liver failure. This suggests that CXCR4 receptors in the MSC could be target for improved MSC-based therapy for end-stage liver disease. The secretion of trophic factors is another proposed explanation for the regenerative effects of MSCs. MSCs were initially thought to exert its effects solely through migration and direct transplantation to the site of injury. However, studies demonstrated suboptimal migration and engraftment, and the newly proposed explanation was that MSCs elicit their effects through secretion of trophic factors. These factors include growth factors like hepatocyte growth factor, vascular endothelial growth factor, or insulin-like growth factors, cytokines like IL-10, and chemokines that stimulate angiogenesis and tissue regeneration. In fact, studies have suggested that extracellular vesicles containing these trophic factors can be isolated, and these acellular vesicles suppress toxin-induced hepatocyte apoptosis, reduce inflammation, and fibrosis. Isolation of these extracellular vesicles may yield an acellular approach to therapy with decreased risk of tumor genesis and fibrosis, usually associated with direct stem cell transplantation. MSCs also have implications in treatment of hepatic fibrosis. MSCs can upregulate the expression of various matrix metalloproteinases, which degrade the extracellular matrix and reduce fibrosis. Furthermore, MSCs downregulate tissue inhibitors of MMPs to augment this effect. MSCs also indirectly inhibit the activation of hepatic stellate cells through various trophic factors and thus decrease collagen synthesis. The multifaceted antifibrotic mechanism of MSCs is a compelling therapeutic target for the reversal of hepatic fibrosis. There are many ongoing clinical trials of MSC-based therapy for liver disease. However, there is not yet a consensus on the optimal approach to treatment in terms of harvesting, administration, or dosing. However, it is clear that MSCs have many properties that make it a great target for future research. There are exciting new developments in the field, such as the capacity of MSCs to recellularize 3D architecture of decellularized liver and maintain viability, implying the possibility of synthesizing a transplantable liver in vitro. In conclusion, MSCs have vast potential in the treatment of liver pathology of all etiologies, and future studies in optimizing MSC-based treatment may create a novel approach to treating end-stage liver disease.